It's just a short story, and it began with looking at 16S trees, which I do a lot of, and there are a couple of basal lineages to the cyanobacteria. So here are the cyanobacteria in green. Can you see my mouth? There we are. And these are environmental clusters that are monophyletic with known photosynthetic cyanobacteria. And I was interested, and several other groups were interested in finding out about what these organisms did, because you find a lot of them in aphotic systems. So then the hypothesis would be that these could be representing non-photosynthetic cyanobacteria. So there was a very nice paper published in 2013 from Ruth Lay's lab, Di Renzi et al. And here's a picture from that paper. And these are all environmental clones. What surprised me was I was looking through the database, and this is a Green Genes released 2013. And in amongst one of these environmental lineages, which is just called SM1D11, there looked like an isolate. And I was very surprised, because I thought, what is that doing there? And it had a great name too, Vampira vibrio. And my first instinct was it was a mislabeled clone sequence. So we started to dig in and look, well, where did this 16S sequence come from, and what was its history? The 16S sequence came from the RefSeq's targeted loci project. I contacted Tim Lilburn and about this sequence, the HM038,000. And it was from actually a deposited culture, and they had sequenced the 16S directly off lyophilized material from one of the ampules. And the reason they did that was because they couldn't resuscitate it. They couldn't grow it, so they just sequenced the 16S directly off it. So we went back to the literature, and it was originally isolated in a Ukrainian freshwater reservoir in 1972 by Russian researchers. And these are they did some nice electron micrography work also in the following years. And it shows these tiny little vibrioid cells attached to microalgae, chlorella. And they're very small, 0.3 to 0.5 microns in size, and the host that they target is Chlorella vulgaris. Now, it was originally classified as a Del vibrio because it's a vibrio shape, and also because it's a predator. But it didn't do what Del vibrio do. So Del vibrio invade their host cells. They get right inside. Whereas these ones really are like vampires. They latch on to the outside, and they basically suck the contents dry. Hence the name Vampira vibrio, which was coined in 1980. So here we see you co-culture them enough. After about 5 days, the algae start to look very anemic, and then they become shells, and then the cycle repeats itself. So we went to try and get one of these ampules, and we managed to locate one from Scotland with the NCIB 11384. See, it's deposited there on 16th of February 1978. And it's green because it's a co-culture of the prey cells and the Vampira vibrio. So there was much heated discussion in the lab about what we would do with this precious ampule. And some of the people wanted to culture it. And we asked around. We knew that the ATCC hadn't been able to grow it again. Now, that could have just been because the ATCC didn't do their ampules right. No offense to the ATCC. We thought maybe this one will be right. But then we asked a bit further, and we found out that a couple of groups, one at Caltech, Woody Fisher's group, had tried to grow it and without success. And I said, they got the 16S sequence. Let's just sequence the ampule directly and see if we can get some of the genome back. By the way, this is a micrograph. We took a little bit to resuspend it. So we saw the Vampira vibrio cells. We saw some intact chlorella, and then you see the shells of the ones that had been sucked dry. So we did that. We got a pretty modest amount of sequence back from MySeq, a little under a gig of data. And we assembled that into 112 contigs with a total length of 3.2 million bases. And when you put that on a chart showing the coverage versus the GC content, you got a nice high coverage cluster of contigs that stood out from the background. And we could go on to show that this indeed was the near complete genome of Vampira vibrio. So its GC content being about 50%, and we got it back in 2 contigs, of which we could determine that a couple of those were plasmids. They were circular plasmids. And we estimated the completeness at pretty close to 100% and a little bit of contamination. Now, these low these low coverage contexts here are coming from the chlorella. So what that was saying is that the vampire vibrio did their job very well. They digested the host, so we didn't really have to fight very much with the host DNA to get the genome back. So the first thing we did was to check to see if that, I forgot to mention that, in the record here, they had been still classified as delta proteobacteria. So they sequenced the gene, but they didn't do the comparative analysis. So I wanted to check that that wasn't actually a del vibrio, and we made a genome tree, and sure enough, it fell out exactly where you would expect, basal to the photosynthetic cyanobacteria, and in amongst these other genomes that had been recovered as part of the Durienzi study and some that we had obtained. And that group is called the melanobacteria, by the way. So we did a metabolic reconstruction of the genome, and the important parts that I wanted for this talk was that we identified flagella. Obviously, you need to be motile if you're going to sense out your prey and attach to it. And they had a lot of two-component sensory systems, chemotaxis, and when we looked at the domain structure of those MCP genes, there were a couple things that we could tag down to what they might be sensing. So they had a globin-coupled sensor, and that's been shown previously in other organisms to be for aerotaxis, to sense oxygen. And then there was also a light-activated kinase. So if I'm a bacterium and I'm targeting a microalga, being able to sense gradients of light and oxygen are a good idea if you want to try and find out where the microalga might be. So that made sense. They had a good suite of hydrolytic enzymes, because if you're going to digest the contents of that microalga, you've got to have some armory to do that. So they had 100 or so proteases, 120 so carbohydrate active enzymes, and then they also had an alginate lyase, which is a pretty common component of algae. So that made sense too. So then you've got the mechanism for finding your host, you've got the mechanisms for digesting it, you just need the thing to lock on and do a docking station. And they also had a T-type conjugation type 4 secretion system. And so they actually have 3 copies of this. And this is the same family, the T-type conjugation system, as Agrobacterium tumefaciens, which obviously is well known for its ability to interact with, sorry, not to digest it, but to send its apparatus in and start co-opting the host cell. So those plasmids I mentioned, 2 of the 3 copies of the type 4 secretion system were on the plasmids. So that's the genes in red. So you had full T4SS operons in here. And I was hoping that maybe we see some hydrolytic enzymes on these plasmids, which we did not see. They were all chromosomally encoded. But in addition, they had a bunch of efflux pumps encoded on the plasmids. So our working hypothesis then is that you have the plasmid, you express the T strand with a relaxase, and then you port it across to your host cell, which is attached via the type 4 secretion system. So this type of secretion system can export both single-stranded DNA and proteins. So we think it's shuttling across some of those genes on the plasmid into the prey cell, as well as the hydrolytic enzymes being transferred across. So then that gets incorporated into the prey cell and starts to express, and that's then locks in some exporters into the host cell and starts to digest the contents, and then transports across the lysates from the algae prey, and it's imported back into the attached vampire vibrio. Or actually, it would be able to, a nearby vampire vibrio would also benefit from this type of mechanism. So then, just in conclusion, this expands the number of bacteriophyla with predatory representatives to 5. And we're interested to know how extensive this predation might be within the lineage, because we just have the one representative so far, because at some point then they will have to have evolved this mechanism. It further supports the assertion that this whole group is non-photosynthetic. There wasn't any evidence for any photosynthetic machinery in this organism either. And there are other components which are in that genome which are relevant to looking at the origins of photosynthesis. And finally, it also indicates that maybe direct shotgun sequencing of lyophilized material might be a viable option for getting back genomes of these non-resuscitatable bacteria. And actually, this is a fairly big problem in culture collections, like certain groups which are very difficult to grow in the first place. You lyophilize them, they go into the culture collection, and then nobody can ever resuscitate them. So planktomycetes are a very good example of this. It's very hard to resuscitate planktomycetes. So this might be a viable option for getting back the genomes. Okay, so just in acknowledgements, this was the work of a PhD student, Rochelle Su, by and large, and some other collaborators at the center. And we thank the culture collection folks for bearing with us and digging through their backlogs to find it. And the study was funded by the Australian Research Council. Thanks for your time, and I'll take any questions.
All right, we have time for a few questions. No one, everyone's looking to go to lunch. So are there any, do you think anyone will be able to successfully culture this now that they kind of know what it is, or do you know where to find it?
Yeah, you can find it in freshwater lakes. So we did try and do some isolations, but not with any success so far. I think as far as that culture goes, though, nobody's going to be able to resuscitate that one.
It's done. All right, yeah, Rhoda.
So would you think that this vampire vibrio without the plasmids will be not predatory?
Yeah, that would be the prediction. It's not going to be able to do the job. Well, it does have one chromosomally encoded type 4 secretion system, so it has yeah, it may still be able to do it. It'd be interesting to find out.
When all these degrading, enzymes that you found are found on the chromosome, right?
Yeah.
This is indicative that the lifestyle of this bacterium is still predatory. Also, vibrio doesn't have a secretion system, still enables to...
Yeah, but it has a completely different mechanism. It gets inside the cell, fully inside the cell, so it doesn't attach. It doesn't have this attachment process. Yeah.
Oh, very interesting.
All right, it looks like we have one more.
I was just wondering if you thought that if you synthesize these plasmids and put them in vibrio, if you could reconstitute the vampire vibrio.
Into vibrio. It's a nice idea, but I very much doubt it would work outside the context of its host.
Oh, wait, George.
Phil, do you know what the cryoprotect was that NCIMT uses?
No, I don't.
Because doesn't ATCC uses skim milk, right? And would you run into problems if you're trying to amplify out of skim milk an entire genome?
Well, they got the 16S back. We know that much. So it was you could amplify a single gene off. I think that Woody Fisher's group tried to do the same thing off the ATCC lyophilized material and weren't successful. So that may be something to look into. Yeah.
All right, well, thanks, everyone. We'll